When the genetic code of our DNA was discovered, our world changed. We had identified the biological blueprint that defines everything about who we are. From there, the technology to explore our genome evolved rapidly. Programmable DNA editors like zinc finger nucleases and TALENs could change specific genes, and RNA interference could prevent gene expression. These tools transformed us from genome explorers to genome engineers. We could not only observe, but also change genes and investigate their impacts to understand how our biological blueprint is read. The advent of CRISPR-Cas9 technology brought versatility, expanding the engineer's toolbox to include virtually any type of change, from gene silencing to custom sequence insertion to transient activation or inhibition. Anything is possible. The efficiency of these methods allows us to explore and engineer on previously unimaginable scales, studying entire genomes and gene expression in single cells. Genome engineering has even entered the clinic, initiating an exciting new era of personalized medicine. CRISPR can generate chimeric antigen receptor T cells that target cancers like leukemias and lymphomas, and gene therapies to repair damaged genes are being tested to treat diseases. Our team is here to make all your genome engineering ideas possible with expertly optimized tools to create any type of modification at any scale. We provide more than just reagents. Our scientists also partner with you, sharing their knowledge through technical support to help you take those ideas from possibility to reality. The future of genome engineering is filled with the potential to transform medicine, and we look forward to building the foundations of better health with you.